separate. There are people who have. Test.
ये एक भी नहीं मिलेगा.
All right, I think we are finally ready.
हमेशा.
We are almost there. Can people in Zoom hear us? Can anyone from Zoom confirm that you can hear us, you can see us, you can see the slides?
We can hear you.
Okay, perfect. Okay, so welcome back after a long holiday. Hopefully you enjoy it. Hopefully you have a great, wonderful Thanksgiving holiday with the family and everyone. So it's really great to see you again. We have about 1 week for the entire semester to end. That's amazing, right? All right, so we'll start now with 2 talks on genomics. Hopefully you already got introduced to one of the lecture, I think the last lecture, right? And we'll start with Jahin Al with Jahin and his colleague. I guess you're ready, right?
Yeah.
I have a microphone. All right. Yeah, we can start. Go ahead, please.
So hello and good evening, everyone. So first of all, we would thank for professor for giving us the opportunity to present this paper. My name is Vansh Thakkar and my teammate Jahin. We are going to present the research paper based on green filter fast seed location filtering in DNA read mapping using processing in memory technologies, which was published in the conference BMC Genomics volume 19 under article 89 in the year 2018. So we'll start. So finally, holidays over. Welcome back from the Thanksgiving. And I know half of you are still in the vacation mode, including me. So more, yeah, almost all. So is anyone here who are still in vacation mode? That's weird. Then I'm not alone right now. So one more thing, don't get hungry after seeing this turkey because we haven't brought any, because we don't have a good budget. And second thing, we are not a good chef like Mansoor, but we have brought some ready-made snacks, Doritos. So let's start our presentation.
So if you have a turkey in the box.
Okay, so first we'll start with the executive summary. Our paper is mostly focused on the genomics, right? So genomics is like study of all genes in our body. So now we'll start with the problem. The main problem of this paper is read mapping. It wastes a lot of time because DNA reads, but DNA maps every read may match millions of wrong places in the genome. So after that, we'll go with the core idea. The core idea of this paper is like it introduces the green filter quickly removes this wrong location before the alignment happens by checking every bit vector. Now about the architecture in this paper, so it is implemented inside the 3D stack memory. We will go deep about this topic in the upcoming slide. Now we'll go to the key results. So key results are it reduces the number of candidates and it speeds up the process. It speeds up the reading map pipeline. Now the takeaway is like it drastically accelerates read mapping by reducing the energy and integrates easily into the existing genomic pipelines. Okay, now before shifting to the technical term, let's start something with simple. So do you know what's happening in this image, or what kind of image is this? Anyone?
What do you see?
What do you see in this image? Yeah, yeah, that's correct. This is an airport security checkpoint where every passenger has to pass through this before boarding the flight, right? So here's the interesting thing. But in this filter, not everyone passes through easily. For example, one of my friends, Ritesh, is carrying knife in his bag and is passing through the security checkpoint. But he would be stopped at further for the further checking, right? And some of the passengers can pass it easily. What if there would be no filter at security checkpoint? What will happen? It will take forever for checking each and every person manually. So this is the exact situation what's happening in genome sequencing. So now we relate with the genomics and bioinformatics. Bioinformatics is where the biology meets with computer science. Think of it like biology goes to the gym and come back as a programmer. So in the scientific, if we want to know in scientific definition, the bioinformatics uses computer technology to first collect the data, stores, analyzes, and analyzes and spread the biological data and information for the further use. Now we will see the genomics. Genomic is a comprehensive study of all the genes, as I mentioned before. It surrounds sequencing an organism's entire DNA and understanding gene function. So imagine if you're sitting in a group of people, and you will see that there will be different. The people around you have different skin color, different hair texture, different body shapes. They all are healthy, they all are normal, yet they all are unique. So here's the first question comes up, like, what makes us different from each other, right? So we'll go one step deeper. For example, let's suppose two persons having the same disease, and they goes to the same doctor and they gave the same medication to both of them. After taking medication, one person heals quickly and the second person doesn't heal quickly. So here comes the second question, is like how diseases and medications also affect us differently, right? So if we go deep into this mystery, we'll find the answer which lies on genomic. Genomic controls everything. It controls our height, the body shape, how we function, how we react to the illness, how we react to the environment and all. So now we'll see, I have shown one picture which tells us, okay, sorry. Now, here is one picture which represents the genomic size of different living organisms. So first we have HIV virus, then we have bacteria, fungus, fly, chicken, rats, humans, and chimpanzee, of course. So these are this is in the increasing order of the genomic size, right? And we should know one thing, that humans have approximately 3.2 billions of base pairs. And I want to ask one more question to you guys. Do anyone know which living organism has the highest genomic size?
Maybe guess.
You can guess the range also. That's also fine.
Here we have for human.
So in this image, you can see this is approximately 3.2 billion is the highest. Yeah, but
Also
chimpanzee is also here, but what
it's
is
more than that? Can you
Yeah.
guess?
Yeah, that's correct. Yeah, there is one Japanese plant which has approximately 150 billion base pairs. That's exactly correct. You can get one Doritos. So here you can see it's a Japanese plant, and the name is very hard to pronounce. I don't know, but yeah, it has approximately 150 billion base pairs. So now we'll go to the next slide. Here comes our main hero of the paper, which is DNA. So DNA, or we can say deoxyribonucleic acid, is a genetic material, and if we found almost in every living organisms. So it stores the biological information which is required for our growth or for development and for our functioning. And it has a double helix polymer of nucleotide bases. It has four types of DNA: A, C, A, T, C, G. A stands for adenine, thymine, cytosine, and guanine. And there are some basic pairing rules, like A always pair with T and C always pairs with G, right? So we have here one 3D image of DNA, how DNA looks, right? Now we'll go further. Genomic research works on genomic research works on two major environments. One is the wet lab, and the second one is the dry lab. Wet lab is where the physical biological work happens. So first we will start with sample collection. We'll collect the sample of DNA. How we'll collect the sample of DNA? Like we can get from the blood or from the saliva or from the tissues of all the living organisms, right? And after collecting the sample, we'll hand it over to the scientist or for the lab preparation. In lab preparation, the DNA will be extracted, it would be purified, and after that, it would be chopped into the small fragments. And after chopping this into small fragment, we will give it to the sequencing machine. So this is the whole process of wet lab. So here is simple analogy to understand it. Think of it like you are cooking food in the kitchen. So what would be the steps? Like first you have to collect the ingredients, we have to prepare it, and we have to cut the ingredients, and after that we'll cook it. And at last we'll find our food, right? So here is this exactly what's happening in here, but instead of food, we are getting digital data from the sequencing. Now we'll come to the dry lab. Dry lab is where it handles computational operations on dry lab. So after getting the raw sequencing data from the sequencing machine, we will convert it into the reads, or we convert it into string as shown in the picture, like A, G, C. This is one read we have converted from the raw sequencing data. And what we'll do with this read, we'll compare this read with the reference genomic in the genomic analysis. So find where the piece belongs, right? So I want to ask one question in this. What do you think of which of this lab takes more time? Wet lab? You should also say why, not guessing on. But don't you think
Computational we can increase with some.
I have also mentioned right now that human genomic contains approximately 3.2 billions of base pair. So in here, we will get approximately more than 3.2 billions of short reads, because it is converted into the short reads, and each read we have to compare it with the reference genomic.
Okay, so then dry lab next.
With the specific like genomic analysis part is the main.
Yeah, so we have understood that. Yeah, if the machine is not optimized, in that case we can say that the wet lab takes more time. But if the machine is optimized sequence, so we can consider that the dry lab is more. Yeah, so yeah, that's awesome. Mhm. Oh, yeah, I see. Because it's weird, it was mentioned somewhere like if the sequencing machine is optimized, then it would work faster, it would generate faster results.
Remember, there is a man in the
Yeah,
middle of the cloud.
yeah, that's
Again, feel that.
yeah, that's now before moving forward, I just want you guys to see one short video about the sequencing, how it works.
High frequency sound waves break the DNA into smaller about 6
Anyone?
special tags are added to the ends of the fragmented DNA. These tagged strands of DNA can then attach to a glass slide. In a sequencer, each piece of DNA is copied hundreds of thousands of times, which creates clusters of identical DNA fragments. Next, the sequencer reads the DNA one base at a time, using different colored tags for each DNA base. Special sensors within the machine detect the different colored tags. This sequence of colors reveals the DNA sequence of each fragment. Powerful computers piece together these individual DNA fragments and reveal the sequence of your DNA. Then a team of medical experts uses specialized software to analyze and compare the DNA sequences to identify the handful of variants that may be important for your medical care.
Anyone understood anything after seeing this video? What's happening here? Anyone? Sorry, what?
If the animal in DNA.
Yeah, kind of. It's true. So yeah, in sequencing machine, what it will do, sequencing machine are the tool which converts the biological DNA into the digital data. It reads the 4 of letters at A, T, C, G, and it converts it into a short pieces, which is called read. So short reads are cheaper to produce, but it's hard to analyze. But based on the requirements, nowadays we can also get long reads. But you should know one thing before generating, like long reads could cover more genomics, but it may give high error. There is much chance of getting high error rates. So now we'll discuss about multiple sequencing machines which you are using nowadays. So first we'll discuss about the Illumina. Illumina is known for its short read sequencing, and the read length of this Illumina is around 100 to 300 base pairs, and it's widely used in the genomic and clinics. And the second one we'll discuss about the Oxford Nanopore. It is known for its long and short read both, and the read length of this machine is approximately more than 100,000 of base pair, which is much longer. And the third one, we will discuss about the Pacific Bioscience. This machine is known for the long reads, and the read length of this machine is around 10,000 to 60,000 base pair, which is a bit longer than Illumina. It uses in different structure variations, and it's more expensive, and in some cases it gives lower accuracy than the Illuma. So from this, one of my teammate Jahin will continue.
Thank you, Vansh. So we are discussing about different steps of genomics, right? So till now we get to know how sequencing machine works. So are you guys with me, right? So what are the steps? Can you remember one or two of the steps till now discussed by Vansh?
Collecting. First
Yeah,
collecting, then
yeah.
fragmenting.
Yeah,
then
exactly.
sequencing.
Oh, okay.
I have a
So
question.
yeah, sure.
Sequencing machine. What is the input to the machine? Is it like the live DNA or physical DNA? How it is? What is the input to the sequencing machine?
It is you collect the DNA, you collect the samples, then you collect the DNA for the after processing, and you chop it into the
Okay.
small
So is it
fragment.
like live DNA, physical DNA, or
No, it's chopped version of the DNA. Like DNA is a double helix, right? So when you chop it, you'll get a smaller portion of the DNA, and you take only one strand. In the one strand, you'll get like sequence of like A, C, T, G, right? And you give input of that one chopped single strand, and then you get output from the sequencing machine.
Because every cell in our body contains this 3.2 billion genomic. So it's very difficult to analyze all. It's very difficult to analyze all this at once. So we chop the DNA, and after that chopped DNA, we'll give it to sequence.
So this is it actually. From the sequencing machine, what do you get? We get the raw data, right? So you may ask that what will be the like format of these files? Like what do you get? What would be the output of the sequencing machine, right? So we have actually we have shown a reference file of the reference genome. There will be two types of file: reference genome file and the read file. Reference genome file is actually the whole sequence of every organism. So if we check on the reference genome file, we call it a .FASTA file. And for that file, the first line it starts with the greater than sign. It is the name of the that sequence. We call this an identifier, and after that the whole the sequence, you can see the sequence of the letter. This is actually the sequence of that genome. So it contains A, C, T, G, and also if you may also see in some portion N, that is we don't know, we don't actually know what is actually in that position. You can also see that in this type of file. And after that there is the read format file, and this we call this .fastq file. And for this file there you will be seeing that four lines. In the first line you see it start with @ and some letters. This is actually the identifier of this sequence. Then the sequence, second line is the sequence one, and you can see the same letter, 4 letters, and also on the N. We don't know N as in we don't know what letter is there actually. And after that the third line is the with the plus sign, it divides the second and fourth line. Fourth line is actually the quality score. Quality score is this sequence is actually output of the sequence machine, right? So there will be for each machine, each letter it also gives a score value, quality score value. So if you want to guess what is here is 29, right? So for 20 to 30, it will be like this score is calculated in log scale. So 20 to 30, it will give you around more than 95% accuracy. So if it is less than 20, then the accuracy will reduce furthermore. So before that, can you go back? So I have a small question. So can you yeah, can you tell me anyone that why for FASTQ file there is a quality score and there is no quality score for FASTA file? Anybody have any idea? We can see there in the fourth line there is quality score for FASTQ file, right? But there is no quality score for FASTA file.
that is reference genome, right?
Yeah.
Is it the output of the sequencing machine?
Yeah, we can say that. Yeah.
So it would make sense to calculate the quality score for that, but you wouldn't really calculate the quality of that which you're trying to align the reads to. Like the reference is just the reference. That's the big
Yeah. That is correct. The reference genome, you're also correct. It is the reference genome. So the actual yeah, both are there, but not actually correct.
Good in terms of say, organism, like it is nature. Then now we have a fast tool, right? That can differentiate between multiple organisms. And so 100% we're sure about pasta that is easy. But here if you don't know what the pattern process.
But how do you get that FASTA file?
Your organism, then build it as a
Can you look at a DNA and build this file? Yeah. So the thing is, first of all, we get this reference genome also through this all process, right? But how we get the first of all, after the sequencing, we get the raw data, and after that, this go a certain processing, right? Polishing, and after that, combining all this after multiple study, we get this reference genome file, right? And it is getting updated every time. Like for now, maybe human genome it is 99.9999% accurate. Maybe next year it will be, we'll add another 9 to that. So it is getting updated every time. So this is actually polished version. So we can call this as a 100% accurate. We can like almost 100%, but we can call this at 100% accurate. But this is the this is a FASTQ file. FASTQ file actually it is just output of the sequencing machine. There is no processing after that. So this is the raw file. That's why there is a quality score for that machine. But for FASTA file, there is no quality score. All of you guys are always almost there, but I just described it more. Okay. So I have shown you the format. So I'll just show another like analogy. So in the left side there is a full picture, right? Full picture you have. We can consider it as a reference genome. On the right side there is small puzzles. So these puzzles puzzles are like reads, as I shown you. Because we get the reads from after chopping the actual DNA small portion, and through the sequencing machine we get this reads. So these are actually small puzzles of the actual big picture, right? So if we have this reference genome, then it will be easier to solve any puzzle, right? So if you want to check, I have like one small puzzle with me. I wanted to give you this puzzle to solve yourself, but the thing is you'll be more into the puzzle. That's why I finally decided not to give you. So see, we have a puzzle, right? This puzzle. So these puzzles have very little pieces, like 24 to 30 pieces. So if you have this puzzle, and this is already like matched, but if you get the like unpuzzled one, and there is a reference picture, right? So if you see reference picture and try to match it, it will be very like easier to match, right? But if you don't have this picture, like reference picture, what do you think? It will be easier, or how much time it will take? For let's say 30 pieces, it will be like you can easily solve it for less than 5 or 2 minutes, right? But think about 3.2 billion character. How much long it will take? It will take a very long time if you don't have the reference genome. But if you have the reference genome, then it will take less time, but even then it will take a lot of time because you have a lot of characters. Next. So this is what happens actually in read mapping, right? You get the small portion of reads from the sequencing machine, and you need to know that what is the position of that read in the reference, right? Or you also need, maybe you don't know which organisms read that is coming from, right? So in that case also you need to match with multiple reference genome to know which organism's DNA portion is that, right? So that portion mapping the reads with the reference genome. Here it is shown with different colors, like in place of characters. So if you want to map and know what is the location or which organism it is, that step is called actually read mapping. So now I will explain the read mapping step with same two file format I have shown you earlier. This is the FASTA reference genome file and FASTQ file. So in the FASTQ file, this is the read, right? So you will have to match with the reference genome for the whole reference genome, right? First you'll match in the first location. In the first location, you will check each character of the reads, right? So let's say it is 100 letter length of the read, then you have to check 100 character. Then again for the next location, location 2 you have to check. Then again 3, 4. Similar way you have to check all the way through to the reference genome. full reference genome to check, look for the position, because if you get a match in one position, you have to keep checking, because human genome is all replicating. So you can get multiple locations, and based on that location, you can see the positions, and also there will be some error tolerance, like if you want to count 1 or 2 characters error, this type of things comes up. So so if you want to check all the way to different genome, how then how much time it will take? Like you can also go for checking multiple reads, right? You will have if you generate reads from a human genome, you will get millions of reads. So if you want to check millions of reads, then it will take a lot of time. If you think about computational, it will take O of M and K, where M is the read length and N is the length of the reference genome, and K is the number of query, it is the number of reads. So you can imagine how much long it will take if you check all the way one by one later. This is actually called brute force algorithm. So it is actually very computationally heavy. So there, in the next slide, we will discuss about how we can solve this issue. So there is one small. So anyone know what is this? Anyone have any idea? This yellow pages, yellow book? Sorry. Yeah, exactly. So we know there is yellow. We used to call this yellow books. Like it keeps all the phone numbers of all the persons in the city or around. So if you want to find one person's phone number, what do you do? You search for the first letter alphabetically, then you check for the second letter. That this way synchronously you find the name of the person, then you get the phone number, right? So similar technology is used in ReadMapper. We have to check through the reference genome, right? So we do like index, we do the indexing on the reference genome. So this is also called a hash table. So we build a table from the reference genome. We take like a small portion of the reference genome, like 5 character or 4 character, based on your requirement, maybe 10 character or so. We also call that k-mer. K is the number of the character, 10 mers, 7 mers, like this way. So we go through the full reference genome once and check for the position of that small strings. Like let's say in place of for first 10 character, it is shown as the green box. So we will look for that green box. This green box as in that particular sequence, like SCTTG, maybe something like that, one sequence. So we'll look for positions of that particular sequence in whole reference genome, and we'll save the location of that sequence. Likewise, we'll look for all possible sequences in the reference genome and store the position of that sequence in the reference genome. That way, we get to we'll get the hash table, and which will have the position of different sequence. So now that we have the hash table. So now during the read mapping, what we will do, we have the location of different sequence in the reference genome stored in a hash table. So we have FASTQ file. So instead of looking for all the position, first we'll look for the position of the, first we'll get the small seed from FASTQ also, and we'll look for the position of that small sequence in the hash table. Okay, so we'll match with the reference genome only in that position. So let's say for FASTQ we have red, green, and blue, right? So if we for the first red, we'll look for the position. Let's say this is the first red, right? But after the first red, there is no green and blue. Okay, so it is not a match. Then we'll look for the second red. This is the red. After red, there is a green, then there is a blue. So this will be one match. So this way we'll look for the position for this seeds only, not all the way through the reference genome. So this is where if we have the position stored in the hash table, we get to like reduce the computation time. Next. So these are the steps of the read mapping, how we do. And then now we'll discuss about some related works done in past about this. First, it is F index VW2. It is a genome mapper use compress index like Burroughs-Wheeler transformer to quickly find the candidate matches for each read. What it does, like, and this technology is also used by tools like BWA and Booty. They quickly find the candidate location without scanning the whole genome. Then next. Next, the seeds and extend. This algorithm, what it does, it creates a shorter reads and find the location of that seeds in the reference genome and extend the read over that location and match the position. It is these are similar like hash table mapping also. Next, there is also some work going on with hardware acceleration. This what it does, that whatever the read mapper step is and whatever the sequence aligning is, they accelerate the hardware steps. They use different like FPGA and GPUs to accelerate those steps. Next, there is pre-alignment filtering. So in the read mapper with hash table, what we have seen that we get the locations of the seeds from the hash table, right? And we look for matches in those locations. Well, during that time, we also see that there is a lot of mismatch in those locations also. What if we get get to do that redirection of those locations also? That is what pre-alignment filtering does. Before checking those locations, it filters those locations and removes some of the locations and do the pre-alignment filtering. And this is actually what it does in this pre-alignment filtering steps. And there are also some mentioned papers also based on this verse. Next. And based on pre-alignment filtering, our green filter is steps are done, and also there is some hardware acceleration steps also included using 3D stack memory. We'll talk about these things later. So in the read mapping with pre-alignment filtering, this is the same example like previous without the pre-alignment filtering. Next. So what happens here, in between there is another step like pre-alignment filtering. In the previous case, what we did, we look for the position of these seeds in the reference genome, right? But here, before looking for the position, it will go through a pre-alignment filtering and it will remove some of the locations. Let's say after filtering, it finds that these are the like less possibility of finding matches, so it will remove these locations, and then it will look for the matches for the remaining locations only. So in this case, it will be like the alignment will be reduced further, so it will be faster in computational. So this was it about pre-alignment filtering. Next, about our paper, our green filter model will be discussed by Vansh.
Thank you, Jahin. Now we will move forward with our main contribution, which is green filter. So Jahin explained what is read mapping, right? How does it work? So while we perform read mapping, the small reads, sorry. So one small read doesn't come up with the GPX location, right? GPS tag, right? So where it belongs. So it could match in many different places inside the genome because in almost 50% the human genome is repetitive, right? So what happens in this? We may get matches. We may get many multiple matches inside the genome. So what will happen is like we will end up checking the same read again and again in a wrong place. So due to this, what will happen? We'll waste a huge amount of time, energy, and memory bandwidth and a computational power. So here we have shown one picture which explains the green filter. Like in the left, you can see there is one many candidates position there, and after applying the green filter, it would check each candidate and it filter out the best one. and you can see that we have left with only few positions. You can think of it like we are applying for some job interviews. Like there would be thousands and hundred thousands of applications for one position, and the interview process, we can think it's like a filter. What it will do, it will filter out and it will choose only few candidates for that position, right? So exactly what's happening in this green filter. So now there are multiple steps of green filter, how it works. So we'll start with the bin. What we'll do, we'll divide the reference geonomy into a bin, and after dividing, we will generate bit vector for every bin. So after creating the bit vector, we will divide the reads into small pieces, which we called as tokens, and this token will compare with the reference geonomy, and if it gets matched, so we will mark it as 1 and 0. 1 and 0 we'll discuss in the next slide. And after matching, we will see that if that bit vector has passed the threshold limit or not. If the bit vector has passed the threshold limit, so it will go for match and validate, and after that it will go to the memory where all this process is going on. So first we'll discuss about what is bin. So basically green filter divides the reference geonomy into a small fixed pieces, and each pieces we called it as bin. So here we have taken one example, like this is a reference geonomy, and we have divided into a small pieces, like, and we have named it as bin 1, bin 2, bin 3, and bin 4, and so on, and it is indicating with a different color. So now what will happen to this bin? What would be the next step? So we have taken an example of bin 2, for example, and bit vector. Yeah, one more thing before this, I want to just explain it. What is bit vector? So bit vector is nothing but just think of it like it's a barcode made up of zeros and one. What does zero means is like if it's zero, then that means that the token doesn't exist in the bin, and if it's one, then it represents that the token exists in the bin. Now we'll take one small. Now we'll take one token from the reference from the bin, and this all are the all possible combinations of k-mers. We will compare this token with this combination, and if these two matches, we will represent this as one in a bit vector. So if we we have taken this GCATG. If we are finding here, yeah, it's fine, it's matched, then we will write it as 1 over here. We will check this for every token in the bin. For example, now let's second, third. Okay. Yeah, so for every bin, we are checking it into the all the combinations of k-mers, and if we are finding, we are representing it as 1. So I want to ask one thing: what is the size of the bit vector over here? Anyone? Sorry? 5 to the power? No. Yeah, that's correct. Go. Yeah, for this bit vector, the size of 4 raised to power 5. So in general, the formula is 4 raised to power n bits, where n represents the all combinations of k-mers. Now now after generating bit vector, what would be the next step? So after generating bit vector, what we will do, we will count all the ones which have been matched with the token. We will count all the one, and we will check if it has been passes the threshold limit or not. If that bit vector has passes the threshold limit, it will be approved, and it will be directly sent to the read mapper for sequence alignment. And if it's not passes the threshold limit, it will immediately be discarded. Now here's one catch. Now here's one catch. What if green filter loses the true match? if I say there is one read and it has true match, but the green filter outs, it doesn't pass on, then what does it mean? Is it dangerous or not? It is dangerous because green filter may lose the true match. What will happen in that case? No. You can say that. So this scenario is like false positive and false negative. So what happening is here, if what false positive is, if a read is having true match, but the green filter mistakenly rejects it, then it's dangerous. Why? Because we are losing true match. So because of this, the author has designed a threshold formula specifically for avoiding false positive. In the same case, there is one false negative. In what false negative? If the read does not have true match, then also green filter mistakenly passes it and it moves forward, but it is less risky. Why? Because it will further anyways, it will be discarded from the alignment, sequence alignment. Yeah. So now we'll discuss, till now we have discussed beans, bean vector and tokens, everything that works logically, right? But the real power of green filter is from where it is executed, right? It is not running in simple RAM or CPU. It is running in some more advanced, which is called 3D stack memory. Normally we have one DRAM layer in CPU, one DRAM layer which is present in the motherboard, right? So if CPU wants to perform something, so data should travel like a shuttle from memory to CPU, CPU to memory, right? So this back and forth process takes time and consumes energy also. So in that case, what we have done in 3D stack memory, we have stacked multiple. Can you show the diagram? So what we are doing in 3D stack memory, we are it's stacking multiple DRAM layers vertically. Vertically, and at bottom we are the bottom layer is which we call logic layer. It handles commutation operations like the token comparisons and thresholds. All the operations are happening in logic layer. So every horizontal layer contains data like bits or beans or bit vector, right? So now in each layer we have multiple banks which allows us parallel access, right? And the volt we have vertically, we have vertical volts are in the vertical column, which allows the independent path for the data. Sorry. So volt is a vertical column that goes through all the floors. Okay, goes through all the floors, just like an elevator which connects all the floor, right? So here a volt. So volt provides an independent data path for the bank to send all the data to the logic layer. So this is how a 3D stack memory is being built. Now from this, Jahin will continue. Sorry. Last time.
Thank you. For this 3D stack memory, we have we have integrated like how the green filter is actually integrated. I will just add to bunch. Like we have bit vectors, right? So in each banks we will store these bit vectors and with all those bits, and there is multiple banks in each DRAM layer. So in each banks, those bit vector will be stored. And whenever a row buffer is activated, then if it matches whatever it is in that bit vector 1 or 0, it will be passed to the logic layer. And we can do this parallelly based on the capacity of this 3D stack memory. So in the next slide, we'll see what are the specifications of this series stack memory. Here I have shown that multiple generation of HBM series stack memory. So we can see that from like first generation to the third generation, the all the channel numbers is increased, and also per pin rate is increased. But I wanted to show that total number of TSVs. TSVs is the lengths of each layer, from each layer to the logic layer. So in our paper green filter, they have used the HBM2. So you can see that total TSV here is around 5K to 7K. So for them, they for this one, what it is, we can parallelly process like around 5K of beans. Okay, so for the beans, we have to generate those bits, right, and check for the like tokens, whether the tokens is present or not. So we can perform those steps like 5,000K beans at a time. Okay, and the memory footprints is 4 to 8 GB. So we also have to check on the memory footprints, like if the bit vector size is one side that 4 to the power 5, then there will be the number of beans is there. So if we multiply the number of beans with the possible bit vector size, then we'll get the total size of the bit vector of the whole reference genome, and we'll get the memory we need to store. So based on that also, we have we need to check on the memory and design accordingly. This is a like simple picture of the 3D stack memory nowadays to give you an idea. Next we'll discuss about the results. What has shown for the green filter. This is a false negative rate versus the number of bit graph. Here we can see if we increase the number of beans. This is the number of beans. If we increase the number of beans, the false negative rate actually decreases. What it is actually. So if you see after 300, it is almost diminishing. So you can select any values after this based on the memory. So in our paper, they have selected 450 beans that actually suits for 5. They have taken 5 tokens. So it is around 3.8 GB for that that specific number of beans and tokens. So it actually suits our CD stack memory. That's why they have chosen that. But if the length increases, then you need to increase the number of token size, and also based on that you need to design the parameters. Next, this is the bar chart of false negative rate for 10 data set they have performed, and it is compared with a state of the art method, that is fast hash. The light color bar is the fastest filter, and the green deep green is the bar for our filter. So you can see for each error tolerance rate, the false negative rate is very low compared to the fast hash filter. And if you go for high tolerance error tolerance rate also, we get very better result for green filter for false negative. Very low false negative rate. Next, here we are showing that the previous same false negative rate for error tolerance 0.05. We can see we are getting like around 5.6 to 6.4 times reduction in false negative rate. It is if you do average around 6 times of reduction in false negative rate. So compared to the fastest filter. How does it get it? It utilizes more information. Green filter utilizes more information from the beans and reads. That's how it is getting that is 6 times lower false negative rate. Next, another important thing is the performance of the filter. It is also done for all the 10 data sets, 10 type of data sets. And if we compare with the fast hash filter, then we are getting better performance. We are getting performing all this in lower time. and the performance shows that average of 2.1 times better performance than fastest filter. And this performance improvement is achieved by both software and hardware combination, software filter technique, and also hardware implementation of 3D stack memory. Next. So next, we'll show what are the takeaways from this. So first, the pre-alignment filtering is necessary, so it reduces a lot of computation. Then next, and also the threshold mechanism is it keeps if we design the build select the proper threshold limit, then you will be able to discard all the low candidates very easily. Next, then they have used the 3D stack memory, which enables parallel filtering and parallel operation, and also which gives us more bandwidth and lower latency and huge speed up. Next, and finally, with the combination of all these steps, we get the result shows drastical performance improvement for this with this green filter. Next, for the conclusion, if we see on the strengths, I've already said that there is massive reduction in candidate allocation, and also using the 3D stack memory, we have performed parallel operation, and also there is in the 3D stack memory there is also in-memory processing, which also reduces the steps, speed up the process. And finally, the most important is we can call this green filter is a modular one. Why it is? This is actually a pre-processing, in the filtering process happens just before the read map alignment, right? So there are a lot of research going on in speeding up the alignment process and also read mapping steps. So we can easily integrate this filtering step with those words to finally accelerate and get a better result with the combination of these two. Next, for the weakness in the green filter, they have already tested this filter with the short reads. So this was not we can also check. There was no mention about the long reads. What it will happen? There will be long reads and a lot of computation and a lot of problem will arise there. And there in the thresholding, if you tune the threshold, if you set the threshold improperly, then you will get a lot of false negative, which will again increase the computation. And also the bit vector size and memory bandwidth, you have to make a trade-off. If you increase the bit vector size, it will hold large memory. But if you have larger reads, in that case, you have to give the take the bit vector size higher, because you have to choose large number of token size to reduce the memory bandwidth. So there is a trade-off. And finally, for the critics, we've shown 3 papers. First, the shows you on, it was published in 2019. In this model, what they do, they do a sliding window algorithm that detects the common subsequence between common matches between 2 subsequences. And this whole process was also use the FPGA hardware to accelerate the filtering. And next, there is another real PIM system. They have implemented this implemented their model in real processing in memory hardware device. They have shown that this is actually practical, but green filter was performed in a simulator called Romulator. It was not directly implemented in any hardware. And finally, the BIMSA, it was published in 2024, and it actually shows the processing in memory technique for long reads also. So what green filter does in green filter, they have shown for the short reads, but they have performed this for long reads and also hardware technology processing memory. They have shown both of the things. Thanks. This is all. That's all from our side. Thank you for attention. And this is just a joke. You can ask any question after this. There are a lot of there are a lot of chips still remaining. You can ask any questions. Yeah.
Maybe I missed it. What is the purpose of the green filter? Like, how does it make
The main thing is you have to you have read, right? You have to know the position of the read from the reference genome. You are searching the positions, right? So for searching, you have to check through whole the reference genome. Or if you have a hash table, you have some locations and you have to look for that locations. What if you have to look for less locations? Would it be better or worse? Let's say you have 100 locations. Instead of 100 locations, you are checking only 10 locations. It will be better. Like checking 100 times, you are checking only 10 times, right? So that is what green filter is doing. It is reducing the possible checking locations. It removes the bad locations where there is a possibility, there is no possibility of getting the match. That was the first model where you check through all the reference genome. That is the basic way, that is brute force algorithm. You check through all the locations by all the position of the reference genome, and also check each letter whether it matches or not. Time complexity. N was the length of the reference genome, right? So length is 3.2 billion for us. Okay, but for now it will check the possible locations. Maybe it will check for only 100 locations. So it reduces the number of locations it will check. Previously it was 3.2, it will check very less number of locations. It reduces the number of locations. This is the only n part is reduced. The rest of the thing is actually same almost. K is the number of query, the number of times you will check, and m is the length of the read, that is also fixed. So you reduce the number of n, you change the n actually.
And the green filter only does it 5 at a time, because you were just the green filter only, you said it has 4 raised to the power n, right? That is the number of.
That is actually the number of vector.
The number of vectors, right? So
yeah.
Each vector is because for every location you have 4 decisions, right? A, G, B, T.
Yeah.
Right. So the green filter only, that's for 5 locations.
Oh, actually, no. It generates the bit vector. Bit vector is the way of selecting those locations.
Yeah.
So when you generate the bit vector, you take all the possible 5 marks, all the combination of those letters, and check whether it presents the token presents or not. So based on that number, it decides whether it will check in that bin or not. So it discards those bins. So it discards lots of bins based on that threshold value. After that, it gets how many bins it will check and how many locations it will check.
Count the number of matching, and after that matching, it will check with the threshold limit. And if it passes, then only it will move forward for the alignment.
Let's say there is in the bin there is 100 letter, and in the reads there is 100 letter. If these two doesn't match, then those short reads will also not match, right? So there will be less one, so that so it will not pass the threshold. So it will not check that bin. That's how it selects the bin and checks. So if it is actually not matching, then that bin will be discarded.
So does the green filter use more space than just doing the regular pre-alignment filter thing?
Sorry?
Does the green filter use more space than, let's say, previous methods, whereby, let's say, you just check the whole sort of work?
Yeah, you kind of save the time, and it also saves the energy also, because we are already removing half of the bad matches before we are doing extensive alignment with pre-filtering also.
I'm talking about the memory trade-off here. You said it in one of the disadvantages is that it uses more.
Oh, okay. You're talking about the memory space.
Yeah.
It actually memory space actually depends on the also the bit vector. If you choose k-mer size of 7, it will have 4 to the power 7. The number of bit vector will increase. So in that case, it will hold more memory, right? But for short reads, like they have shown like for 100 character long reads, 5 marks is enough. So for 5 marks you get better result. That's why it also stays in the memory range of the HBM2. Now we have HBM3, we can also increase that size, and we can also like analyze for long reads also nowadays. Yeah, sure. Yeah.
Question: what is when you speak to the choice of the payments to search for vectors, and what is the influence? And what is the trade-off?
If you use if you think about like okay, so how to choose this number, like for whether you choose 4 marks or 5 marks or 6 marks, right? That is your question. So if you okay, how it influences. So if it is 4 marks, the bit vector will be how much bit will be there? 4 to the power 4. It is 4 marks. If it is 5 marks, 4 to the power 5. So if you increase the number of the characters in the seeds, it increases the bit vector. See, so if you have to store that bit vector, right? So if you increase the number of the character, it will increase the memory uses. Okay, okay. Does it benefit increasing the read length? Okay, yeah, it all. Yeah. So let's say for 100 character length read, right? If you select 2 marks, let's say 2 marks, just 2 character, so there is a more possibility of getting match, right? 2 character, there's a easy possibility of match. It will look for that combination in the bin size, right? If you use 3 marks, 7 marks, like if you increase the like character of that small token, it will if it is not exactly match, there is a less possibility of getting match. Did you get my point? Like if it is a match, let's consider one read matches with one bin, right? So it will match like most of the characters. So in that case, like if you take 7 marks, 8 marks, whatever it is, it will match. Okay, but if it doesn't match, we have to discard that portion, right? So for that, if we take lower like 2 marks, if it doesn't match, that also will give you more number of ones. Okay, it will even then also it will pass the threshold. That's where the actually trade-off is. So for the longer reads, you have to select long like 7 marks or longer number of strings. For the shorter reads, the shorter sequence is okay. This logic part, like thresholding and also bit vector, this part is all software. But how you implement this, we implemented, it was implemented on 3D stack memory, right? That part is the hardware. Actually, this paper emphasized both software advantages and also the hardware advantages. Both combined gives the better performance.
Threshold is also dependent on the value we are searching.
Threshold?
Yeah, threshold.
No, threshold formula we have already shown. Like the error tolerance. No. Okay, with the KMR, threshold will not change. With the change of the length of the KMR, the values of like number of ones will change. So it will pass threshold. It will pass threshold, then it will be not discarded. It will pass the filter. No, threshold is dependent on the read length and also the error tolerance. If it is you want to find the match, right? You don't want to find what are the lengths are. It depends on read length and error tolerance. How much mismatch you want to accept. It is based on that.
Absolutely, that's a great point.
The matches, the ones, the ones are actually it is checking only the small tokens from read, and it is checking from the bin whether it is matches for those small tokens or not. If you take two small tokens, randomly it will generate one, because there is possibility of more finding match with unmatched bins. N is the n is the n is the number of token actually. Sorry, number of lane of this. What is that? Sequence, small sequence. Because with that small sequence tokens, the token length and the error tolerance is also dependent with that. So in the right picture, you can see if there is only one mismatch, it will impact like four of the k-mers, like 4 mers. If it is n equal to 4, then it will impact 4 seeds. If it is 5, it will impact 5 seeds. So based on that, actually the threshold is determined. So yeah, anyway, the token size is also impacting the threshold value. Yeah, it is actually. Yeah, you are correct. You'll get all the Doritos, I think. False negative. When you match with the reads, if it is matches, then it's okay. If it doesn't match, then we get the false negative. That is false negative. Yeah. If it is not aligning, if it is not a match, then it is false negative. Yeah. If you don't do pre-alignment, there will be false negative, but it will be, the number will be higher because you haven't performed the filtering. If the read doesn't match with the genome, then it is a match. That one is for small seeds, a small portion of the long reads we have. No, small seed 4 character is matching, but it doesn't match for 100 character. That's why the read doesn't match. We are checking the small seeds to get an idea whether this bean will be selected or not. To select the bean, we are checking the small seeds. But finally, when we select the beans, then we check whole reads. Yeah. False negative rate will depend on the threshold. Any more questions? If not, then we have some questions. All this processing in memory. Yeah. There is comparator in the. Yeah, there will be. Obviously, there will be some instructions. Yeah. You can design the logic layer custom design. It was implemented actually in simulator called Remulator. Yeah, but they haven't mentioned about those instruction actually. Okay, then we'll go with our question. With the bean we have already selected. The bean get passed through the threshold value. Bean is a portion of reference genome.
Yeah, we cut the piece. We cut into small pieces.
Yeah.
Yeah.
Equal to that read length, same. That when we select the number of bean, we select such a way that the bean is same length as the read length. Yes. We match
We compare it
for,
with match
with the bean. We have the read, but we have the bean also.
Here is
using the bead vector, we select the beans, but during the matching, we select we match with the beans. Yeah. During the matching, we get go to the bean actual reference portion of the reference genome.
yeah, that's good.
Trying to say we have to take one small piece from the bean, which is called token. So we'll take one token at a time and we'll compare with the bean vector. And if that token matches, then we'll convert it into one. And
String
we'll
of
do-
beat.
Yeah.
We have
So
a
we'll
string
take
of
one
beat for those. Then if the string matches, then
So we'll take one. So we'll take a token and we'll match it with the.
Okay, that's logic. What's happening? Wait. Greater than and then we have this factor, we have this match. Something with what
Yeah.
in the token.
Yeah.
Yeah.
That is just like
Compare.
hash rate. Also, how we would know
How? Oh, the filtering process it perform like perfectly or not. It is based on the threshold value.
have a portion
Yeah.
X. Have all very accurate algorithm. That is like
Yeah.
accurate. Run accurate.
Yeah.
They will tell us how much loyalty will be constant.
Yeah.
They give us like very accurate.
5 differences. Yeah.
Then the filter will tell you, cannot tell you the exact number. It will tell you these differences, that threshold or below.
Threshold or not, yeah,
Meaning telling you it's good
or bad.
And again, how you get the actual output.
If you want to compare, then yeah.
This is how you would know.
Yeah, that's true.
This stated value from the game filter, then you can
Yeah.
okay, we better than the variation. The game filter said no, it's too much.
I was talking about only green filter actually.
This is like if a green should be bit factor has total of 10 values and out of that 6 are matching. So that 4 have the 4 values haven't matched, so that could be a false negative.
No.
So
No.
you cannot tell we get it done.
Ground truth, yeah. This
How
actually in the formula it comes up. If it is like one match, it will generate like 4 zeros, one mismatch for 4 marks. So from the threshold formula, we actually defines that how much mismatch we want to accept. If it is less than that, then if the ones are less than that, we will discard it. If it is greater than that, we will accept it. So the threshold formula itself gives us the way which one is false positive, which one is false negative. So if you want to match it with the real value, you have to like separately get the ground truth and match. Finally, green filter works better or not. Yeah. Yeah. Yes.
That's
Yeah. We can work with long sequences if it will extend the memory uses. In that case, we can split this long sequence into shorter sequences. In that case, the memory uses will be lower. Less.
what do you think about everyone?
Five. Yeah.
Say, this was nothing, right? This was the
Yes. Yes.
one minute. Then think about long sequence.
Long sequence.
It's
5A will be multiple times. Yeah. Getting multiple times. So for same sequence, you'll get multiple ones. Then that will be actually not match, but it will pass through the filter. It will increase the false negative rate.
so much.
Thank you.
What? Is this the first presentation? Yeah.
No.
Okay, no question from our side.
Yeah, they took
Hopefully you'll
Yeah,
understand
they
the
took-
topic.
they took
Now we'll
whole
jump
time.
No break. One thing break. You can have. Okay, no break. Continue directly.
Hello, does this work?
So for those people on Zoom as well, please pay attention. So everyone, if I give you 2 options, which one do you prefer? Send you over email. Should we have 3 talks next lecture? Or should we have it somewhere else? What do you think, everyone? How many of you supporting having 3 talks after tomorrow? Tuesday. Thursday. 1, 2, 3, 4. 3 talks. People in Zoom, please raise your hand.
One time we can't even finish 2 talks.
Raise your hand if you support 3 talks next lecture. Cannot hear me. People in Zoom, can you hear me? How many of you support having 3 talks? Please raise your hand. No, no. No, 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 no. Okay. Not your presentation. Not your presentation. So I sent email about. I didn't tell. All right. So how many of you supporting having the 3rd lecture, the 3rd talk on Monday? On Monday, over Zoom or in person. That's the last chance that we could have any lecture, which is 8. No one. So we'll have 3 lectures, 3 talks on Thursday. All right. And now the question is, when should we start the lecture? You know, we started at 5:30. Should we start it at 5 o'clock or 5:15? How many of you having any lecture or commitment before the lecture time? I normally come late. So how many of you? Oh, you'll be presenting that day, right? They should come in. All right, so we should start it at 5 o'clock, right? This is what we agree on. All right, but what is a must? At 10, starting from 5:30, as usual. But please try to come 5 o'clock if you can. All right, that's cool. So hopefully we agree on that. Who sent it?
So I sent.
You sent, Ryan. Quick. Please try to use the microphone for questions so people on Zoom or YouTube can hear you well. Yeah, probably send it to me only. Please always CC supper, no communication. Right? When you send files, when you send your paper, dated paper, when you send whatever, even attendance absence, always CC supper. Done?
Yeah, we do.
It doesn't.
Yes. First, we have a class, right?
Yeah.
So can't even stay for the 3rd one. And we have the after this class we have seminar. You're taking a seminar, right?
Yeah, so seminar at 7:25. I can leave as late as 7:25.
Yeah, but we have 3 talks on Thursday, so you're definitely gonna. You have to tell him you have a class after, so you need to leave. It's not to miss attendance.
I don't have access. Is this the correct one? Correct? Let's assume it's correct. Corey, your laptops.
This is it.
Okay, send it airdrop. Airdrop it to me.
It's too big.
No, save it
It's a
somewhere.
big
Save it
one,
somewhere near the desktop or download.
I mean, it's. But I can-
Yeah, save it.
Yeah, I know. And then,
It's okay, you can share it in airdrop, even if it's large. Can you say something?
Hello.
hold on to it.
I want to hold the laser index. Where's the laser? Yeah. Okay, cool. Hello, everyone. I'm Grigore. This is Ryan. Our paper is named Aligner D, leveraging in-DRAM computing to accelerate DNA short read alignment. So we were supposed to start with a short background info on DNA short read alignment, but we've already seen that in the previous talk, so we'll try to go through it really fast. So we already learned that living organisms have DNA, humans, viruses, bacteria, and we already know that the DNA is the genetic instructions for all life. And we already know that it's made of 4 bases, A's, T's, C's, and G's, and the sequence matters. So I will just add that the single unit of measurement of DNA is a base pair, because DNA consists of 2 complementary bases. A always pairs with T, and C always pairs with G. So if we know one base, we automatically know the other base. So the unit of measurement is a base pair. So then we have this sequencing process that was already covered in the previous talk. So this is a simplified diagram, and I will just highlight that the input is a physical sample, the output is the data. We get short reads from our sample with the help of sequencing machine. And what we actually get, we get millions of reads. We have millions of reads that come from a massive genome. We already learned that the length of the human genome is 3.2 billion base pairs, and the length of one read can be as low as just 150 base pairs. And all of these millions of reads come from random locations in the genome that we sequence. And besides that, there are some repetitive regions in the genome, and there are some sequencing errors, both in the reads and they can be in the reference. And also there are some insertions and deletions, some regions of complexity. So we have this example with matching puzzles, same as in the previous talk, but yeah, so here I can ask the audience this question. If we take two random people from this class, or just two random people in general, do you know what's the average genome similarity, like in percentages? Are they like 50% similar, 70% similar? Any guesses? 99.9. Yeah, you're right. So, and this is why we use reference genome. We are leveraging this fact that different organisms of the same species have very similar genomes. So we can use one genome as a reference. So this is why we need read alignment. We have this problem that we have many short reads, and we have the solution, the reference genome. We find the location for every read, and we get some biological insights. So read alignment is a very challenging problem because there are some complexity regions in the genome. Also, there are some repetitive regions in the genome. So sometimes we have a read, we don't know which repetitive region this read came from. And the entire thing becomes even more costly as the genome length goes up and the number of reads goes up. So this is a very complex mathematical problem, and a lot of effort is being made to accelerate read alignment. And we will show you 3 different directions how we can accelerate this thing. So the first direction is a hardware direction. So basically we can develop custom hardware that will hardwire certain algorithm for read alignment, certain existing algorithm. So we can design ASICs for read alignment in the same way as people design ASICs for mining Bitcoin. And then the second direction is algorithmic direction. There is still a lot of room for developing new algorithms for read alignment. For example, there is this tool Minimap. It uses minimizers. It selects more informative substrings, which algorithmically reduces the time spent searching. And then the third direction is the data-driven direction, processing in memory or processing near memory. So we place computations directly onto or near DRAM chip to address the notorious memory wall problem when we spend time moving the data from CPU to DRAM. So we can avoid that. So here are some recent papers on each direction. So for hardware acceleration, in this paper, they designed some ASIC accelerator for a specific read alignment algorithm, wave-front alignment, and they use a specific CPU architecture, and they achieved a speed up of up to 1,000 times. Then we have this paper published in 2024 with a new algorithm, hybrid alignment algorithm, combines the speed of a hash table with the efficiency of a suffix tree. So they designed a new algorithm. They managed to process a human genome in just 31 minutes. And then the data-driven acceleration. This is our paper, Alomair D. And the key idea is that we can address the bottleneck like of speed and energy by processing in memory. So now we will go in detail about our paper, but before we do that, we want to introduce this XNOR operation, which appears a lot of time in our paper. So this is a bitwise operation, and the meaning is that you have two inputs. If two inputs are the same, then the output is 1. If two inputs are different, the output is 0. Very simple. So executive summary. The problem is that there is this costly row initialization that prevents processing in memory systems to handle XNOR efficiently. And these overheads reduce performance, energy efficiency. And we can change the design to remove the initialization cost. And we can do that by designing some new architecture. We use, in this paper, they use a 3-transistor XNOR gate and a 12-transistor full adder. And they build it on top of some existing processing and memory architectures like MBIT and DRISA, but they make it more reliable for XNOR operations. So the goal of the paper, again, to address the significant computational and energy bottleneck of the DNA read alignment. And to achieve this goal, we leverage 3 key ideas. So the first idea is we propose an energy efficient, high throughput, and XNOR friendly processing and memory architecture.
Right. So I'm going to talk about more of the specifics on the new hardware architecture. There's a significant focus on gaining a reliable voltage amplification, implementing it such that there are a lot fewer transistors column of any DRAM matrix. And so that makes it area efficient. And there's a significant focus on an increased throughput through the use of these XNOR logic. So I'll kind of just go through the basics of the subarray. So a DRAM subarray has this mechanism called the row decoder here on the left. It receives signals from memory controller through a data bus. The row decoder will translate these signals into particular word lines and bit lines. The word line is the horizontal component which connects all of these. Sorry, I forgot I have all of these animated. That's what I'm showing. The idea is that we have these rows, cells, each are containing data or lack thereof. Sorry, I could speak louder. My bad. Right. Sorry about that. So each of these DRAM cells here are composed of 2 primary components. They have a capacitor and an access transistor. The access transistor acts as a switch where it will either be open or closed depending on whether or not the row decoder has activated the word line to open it. Right. So the idea is there is a drain connected to the access transistor, which connects to this bit line, which is the vertical component going up and down. The bit line is connected to the sense amplifier here that you see in the very bottom row. The sense amplifier will sense a very, very minute voltage differential, and it is through this voltage differential that it is compared against. There is a reference voltage that is stored in the complementary bit line. This is compared to the voltage that is stored in the bit line. It is through this difference that sense amplifier can amplify a particular signal resembling a 0 or 1, and thereby whole memory as a dynamic random access memory operation. So the weakness in the existing approaches. So there is an inherent complexity to performing XOR logic. This means that other architectures have to use a lot more AND operations, OR operations, XOR operations to perform an operation, to perform an XOR operation. The XOR operation is significant in our paper because it is through this particular Boolean operator that we can perform sequence alignment matching in one operation that is otherwise performed through multiple cycles. So the other particular issue with these other models is that they will often have to form significant numbers of row copies for these different memory rows. One of the most popular architectures will have 2 input rows, but have to perform 3 copies of these memory rows to other addresses, and that is both costly in terms of time and in terms of energy. And there's also this issue of having inaccurate voltage differential measurements, where there will be multiple rows in the same word line, in the same bit line, that will have open access transistors. The problem is that charge sharing can occur where the voltage read can be inaccurate because there's charge sort of leaking between the cells. This makes it more optimal for our architecture where we only use a single row. And of course, there's this sort of big data issue where there's our short read alignment required. These are the specific other architectures. So you have these operations like a triple row activation. That is what I was just referring to. We have Teresa 3 transistor, 1 capacitor model, and it's a 1 transistor, 1 capacitor model. This last model actually happens to be the closest to performance to the architecture that we're presenting today. This is what they kind of look like. They're very similar to the, you can see some of the same sort of notation in these diagrams. I won't go through each and every one of them, but the idea is, especially with this one on the far right, it has an upgraded sense amplifier, which is attached as a peripheral circuit to the sense amplifier. It has a latch and it has a logic gate. This allows it to perform a lot more local memory operations, local memory subarray operations that are able to increase its speed of performance drastically. So again, this problem is that the existing architectures have different transistors that are more ideal in projecting particular signals. So these are metal oxide semiconductors. We have 2 different kinds. There's the positive metal oxide semiconductor and the negative metal oxide semiconductor. One is meant to, the negative one is meant to project logical ones. The positive one is meant to project logical ones. The negative one is meant to project zeros. The issue with this is when these inaccurate signals are projected to the sense amplifier, it takes longer for the sense amplifier to form its final amplified signal, or to project its final amplified signal as the data read. And these the signals it's projecting are also very inaccurate, or can be inaccurate. And as more Boolean operations are performed successively, these the signals, the quality of these signal assessments degrades each time for concurrent or consecutive in-memory operations. So this is kind of an example. So we have, this is called the fusion input cell. We have that positive metal oxide semiconductor that will be connected to this P terminal and the -1, the N terminal. So as a concrete example, we have, consider row 4, which is performing this logical AND between these two inputs A and B. We'll have this, so for example, if we set if A is equal to 0, that by convention, that will automatically trigger the positive, the PMOS switch on. So if the PMOS switch goes on, then it will automatically, without even assessing B, it will return a 0. PMOS is not meant to deliver a signal of 0, so the voltage that is being read by the sense amplifier will naturally be slightly above 0. That is problematic and leads to unreliable signal inputs between different consecutive operations. So the solution in this paper is using this 3-transistor XNOR logic gate, specifically designed form XNOR. So it uses 2 NMOS transistors. Each are connected to one of the input signals. There's a 3rd transistor that is PMOS. That is where the source of that transistor is connected to a power supply, and its gate is connected to a bias voltage source. So by implementing it this way, and I'll kind of go into more detail on how it works, by implementing it this way, only stronger signals are delivered. This is what it looks like. So there'd be like an NMOS over here, an NMOS transistor over here, and then this is what our PMOS looks like. So the idea is that if there are two negative, or if there are 2 zeros inputted, then both of the NMOS transistors will turn on. Both NMOS transistors will be turned off. The PMOS will automatically turn on and deliver a stronger signal from its bias voltage source. If there's a 0 and 1, then one of the NMOS transistors will be turned on automatically, deliver a strong 0. And if they're both equal to 1, then the PMOS transistor will automatically turn on and deliver 1. So now I'll kind of describe the second component of the nuance in their architecture, which is this full adder. The full adder is a binary addition mechanism. It takes 3 inputs, that being the 2 input bits, an A and B, and then a carry-in bit. Then an output, it has 2 outputs, with that being the sum, and then carry output. So the idea is it's going to perform this XNOR operation between the first 2 inputs and then the carry-in bit. This is a much more complicated operation here. But the idea is it has to consider this carry-in bit and then construct the next carry-out bit, because the carry-out is what will be used for the input as the next and the sum for the next operation. You could think of it in the same way that you would do addition between two-digit numbers, and you're like adding 2 digits from the ones place, and you have to have a carryover. Same idea. This is what it kind of looks like. Right, so you have that, this is like the carry-in bit, translates to a carry-out bit that functions as the carry-in bit for the next full add, and so on and so forth. In this way, you can perform additions for very long sequences of binary values. Right, so this is what it looks like in the implementation. So you might recall before there was that sum operation, which was performed between between the A, B, and C in as 2 separate XOR operations. There are 3 transistors here. This is performing that first sum, the first XNOR, and then this transistor here is added this set of 3 transistors is performing the XNOR between the result of this and the carry-in bit. This is a 6-block transistor. So in total, you have 6, 3, and 3. That's 12. That's why it's a 12 transistor full adder. This will perform all of the logic required to produce the carry out bit. Right. And this here is the transient wave waveforms, the simulated transient waveforms. Very quickly, I mean, so I probably don't need to go into all the details, but the idea is if you have these particular inputs, and for the A, the B, and the carry-in bit, some will naturally translate into values seen at each point. So yeah, the idea is it saves about 65% of the area over traditional 34 transistor model. The number of transistors per column is significantly reduced to perform XOR operations from 14.5 to 5. We have much stronger signals delivered, and we now no longer need to activate multiple rows because the XOR operation can, simply requires one row to be amplified, along with, it's the same idea with the the 12 transistor full adder. More details on the specifics of that operation and why we don't need more rows will follow, but it is because of this 3 transistor logic gate that we are able to accomplish.
Right, so that was the LineRD architecture, and now we'll move to the second key idea, which is the updated algorithm that leverages that architecture. And before we move to the updated algorithm, we will talk about how we actually align reads to the reference. So this is the incorrect approach. If we want to align a read, then we just simply check every spot. So consider that this is the read, right? And we have like 3 billion letters. So this will take like 3 billion comparisons. This is not efficient at all. So we need to introduce some kind of intelligent algorithm similar to how your phone is looking for contacts. When you enter just one character, it immediately narrows down the search to only names that start with that character. So we will try to implement an intelligent algorithm that is based on some kind of sorting. So the algorithm is Burrows-Wheeler transform and FM index search. So not to confuse these two things, Burrows-Wheeler transform is just a transformation of the original sequence. So we have our reference genome. It's a very long sequence of characters, and we can just rearrange the characters, get this Burrows-Wheeler transform. And then we use the FM index search, which is the actual search algorithm that is used to search a pattern in a string. And it is widely used by state-of-the-art read alignment tools. And yeah, this Burrows-Wheeler transform and FM index also was mentioned in the previous talk. So now we'll try to understand how it works. So Burrows-Wheeler transform, the first step is we need to generate and sort all rotations of our string. And this is the core step that helps us group together similar contexts before we can search something. So consider that this is our read, this is our sequence, and we simply build this table of suffixes where each suffix is the substring that starts at a given index. So if you look at the first row in the table and the sequence, this is the entire sequence, because if you start at index 0, the suffix would be the entire sequence. But if you start at index 2, the suffix would be starting whatever starts at index 2. And this way we build this suffix table. Then we take what we got at this step and simply sort it in alphabetical order. The only thing here is that the $sign goes first. The $sign goes before A in alphabetical order. And then we simply keep the indices. So the $sign was 5, so we record 5. And then we keep the first character for each suffix. So this starts with $sign, this starts with A, this starts with C, this starts with G. Then the third step, we create a string by taking the last character of every row in the sorted matrix. So why we do it? Because we want to cluster identical characters together. So this is the sequence. We need the first letter of each sorted suffix. And so we take our F column and we ask a question, what character comes before this suffix in the sequence? So we look at the first character. This is our $sign in the sequence. And then we just simply take whatever goes before that character. And this way we build the last column. So just like that, the last column is nothing else but our Burroughs-Wheeler transformation. So if you look at the original sequence and the Burroughs-Wheeler transformation, we didn't add any new characters. We just transformed, like rearranged the sequence. So now we move to FM index search. The goal of the search is to take our Burroughs-Wheeler transform and find a range that contains our pattern that we are looking at, our read in the sequence. And we do that by continuously shrinking the range. So I will show you an overview of the algorithm, like very simplified. So this is our pattern, our read, and our sequence. I mean, Burroughs-Wheeler transformation. And first, we initialize the widest range. Second, we iteratively compute the new range using the formula. And third, we check the range. If low is greater than the high, then the pattern doesn't exist. So you may notice that this formula actually uses some new symbols that I haven't introduced yet. So to compute the new range, we need to build something called occurrence table and counts table. So let's try to understand it on a very tiny example. So we take our Burroughs-Wheeler transform and we ask a question, how many times HTTG have appeared up to and including row I? So row I is the first character. And then we just simply store how many A's have appeared before A, including A, just one, because we count A. And then how many T's appeared before the first character A? 0. How many C's appeared before character A? 0. And just like that, we build the occurrence table. Then we take our Burroughs-Wheeler transform again, and we ask another question. How many characters are alphabetically smaller than X? So X is the character, and this is the order, the alphabetical order where $sign is the smallest character. So for our example, it's very straightforward. We just simply record how many characters are bigger. So the smallest character is X. There's nothing smaller than X. The smallest character is the $sign, the end of the string symbol. And there is no characters strictly smaller than our X. And for A, there is this $sign. For C, there is $sign and A. So just like that, we built the counts table. And now we actually start searching. So the first step, we initialize the algorithm and we initialize the range. So why we process the read backwards? Why we start with A? Because we have to go backwards because we set up the columns in such a way that the letter in the L column tells you where to go in the F column to find the character that comes before it in the word. So this is, and this is our suffix array, our indices for our suffixes. And we just simply take the entire range first. We take the widest range. Low is 0, high is 5, because we have a total of 6 suffixes. So the lowest index is 0, the highest index is 5. So we initialize the algorithm, and now we move to the second step. We compute the new low and new high. And why we use these two tables because the counts table provides the base starting point for any occurrence of that character when we look at the sorted F column, and the occurrence determines the offset, the shift from the base. And this is our counts table, this is our occurrence table, and we simply look at the column, we look at our character of interest. So in our case, it's A, and we observe that also we look at the occurrence table, we look for A, and we simply take the values from the occurrence table and the counts table. So for the occurrence table, for the new low, we have the occurrence of column A at index -1, but -1 is not in the table, so we just simply put 0. So 1 + 0 = 1. And in the same way, we look at the table and we compute the new high. So we computed the range for A, which is 1 and 1. And then we move to the next character. I will not explain the calculations here, but the important point is that every other step is based on whatever low and high we got in the previous step. So this way, we now process AT, these two letters, and we get the new low and new high. Then we move to the third character, and now we are processing ATC, and we compute the new low and new high in the same way, and we get this interval 2 and 2. So now we move to the third step, and we assess this interval. What does it actually mean that we got 2 and 2? It means that all suffixes between rows 2 and 2 start with CTA. So if we look in our table, yes, indeed, there is this suffix CTA. If you look at the original sequence, this CTA starts at index number 2. So just like that, we found the read CTA in the sequence EG CTA. And so that was the default, the normal FM index search and Burr-Wheeler transform. So now, what was the novel thing that introduced in this paper? So this is the overview of the updated algorithm. So what is important here? We just saw a very small example, and our occurrence table was really small. But if you look at this example, occurrence table can be really heavy, 45 gigabytes and of memory. And we can reduce this space by sampling occurrence table. We can just say, we will take the row number with index 0, and then we will take the row with index 128, and then we'll take the row with index 256. And this way, we will reduce the occurrence table by a factor of D. For example, in this image, D is equal to 128. And you may ask, how can we do that if we are just throwing away information from the occurrence table? So to restore the information, we introduce this new data structure, which is the marker table. And marker table will allow us to restore whatever we threw away from the occurrence table. And how do we do that? So before we move to the algorithm, we do sampling. So sample the occurrence table. Consider our example and just take sampling distance of 2. So then we just take the row number 0, 2, 4. And just like that, we get the sampled occurrence table. Then we take our two tables, sampled occurrence table and counts table, and we compute the markers table using this formula. So we'll look at our row A, and then we'll look at column A, and then we'll look at count. And we compute this marker based on our tables. So if you look at this value, it is computed based on the count of A and the occurrence of A using this formula. And notice that this formula is similar to whatever we showed before with the new low and new high. And this is exactly why we need this marker table, because we want to restore the information that is needed for the algorithm later. So, and now we perform the updated FM index search. This is our table, indices 0 to 4, and let's say we want to find occurrence of character A at index 3. We don't have it in the occurrence table because we only took indices 0, 2, and 4. So it means that if we can compute occurrence of character A at index 3, it means that we can compute any new low and any new high, because we can compute whatever is not in our table, right? So nothing prevents us from computing new low and new high. So how do we compute something that is not in the occurrence table? Step 1, we identify the checkpoint. Checkpoint is just the closest integer value to 3 that is lower than 3, which is 2, which means that we take the checkpoint for row number 2, and in our table, it's checkpoint 1. Then we simply read the marker from the table. The marker is 2. Then we compute the difference between row 2 and row 3, because we need the occurrence of A at index 3, but the closest checkpoint is for A at index 2. So how we compute the delta? We look at our Burr-Wheeler transformation, also known as last column L. And we take this character, we look at this character in the index 3, and we ask, is that an A? No, this is not an A. So this is the step that is done by the new XNOR circuit. This is where we leverage the new architectural novelty, because the physical meaning of this delta is that we need to compute how many A's appear between the checkpoint and our row of interest. And we can do that with the help of our hardware by running this XNOR operation just as many times as we need, as the distance between the checkpoint and our row. So for our example, delta is 0, which means that now we can reconstruct the occurrence by simply adding the delta to the occurrence of A at checkpoint. And just like that, we get occurrence of A3 equal to occurrence of A at position 2. And now what happens if we want to allow some mismatches? So consider that we have this read that doesn't actually match the reference. We start processing it. We start processing it, and we observe that the interval is 3 to 2, which means that the low is greater than the high, which means that this interval doesn't work and there is no direct match. So then we can introduce a new variable, and this will be the number of mismatches that we want to allow. And if we get an interval that doesn't work, we simply compute the interval for all characters, and whatever character works will be our interval, and we make our Z equal to 0 because we don't want to take any more mismatches, right? So we can set this Z to any number. If a character, we are leveraging this idea that if a character doesn't match, then the low is always bigger than the high.
Right. So now I'm going to talk about the parallelized nature of our new architecture. So a lot of implementations, particularly like that, like CPUs, are kind of suffering from this von Neumann bottleneck. Our architecture is not, because we don't actually perform any off-chip memory pulls. So the idea is that we can, using our 3-transistor XNOR logic gate based 12 transistor adder, we can sort of eke out every bit of performance. We're only using one row at a time, so we're not performing any additional row copies, and this will kind of be made clear. So a very special part of this paper is the way that we partition our data. So the idea is where every single memory subarray has 256 rows and 128 columns. The first 128 rows are dedicated to the Burroughs-Wheeler transform data. The second 128 rows hold the marker table values. The idea here is that this arrangement of the data allows for all of our in-memory computations to be done entirely locally. This drastically speeds up our throughput. So the idea is that we have these correlated regions specifically. The reason that we can perform all these memory operations locally is because the regions from the Burroughs-Wheeler transform and our marker table values are specifically arranged according to their relation to each other. So to perform the 12 transistor add, which is kind of the bulk of the throughput, there is a component of the architecture there. It's just called the controller. It's not the memory controller, it's a specific controller. It will receive, it has like an input. It'll be the particular nucleotide of interest from the short read, and then it'll be a range of indices for the Burroughs-Wheeler transform, which will be translated into word and bitline addresses that will be projected to, or that will be accessed by the sense amplifier. So the idea then is that we only need this singular row to get the Burroughs-Wheeler transform. The nucleotide is stored in the buffer. The buffer can be accessed by the 3 transistor XNOR logic gate immediately after sense amplification of the Burroughs-Wheeler transform sequence. The XNOR is performed. A counter inside of the controller is incremented, and it is all these counts that will be stored in the buffer, along with the marker table addresses that are corresponding to the transform. So the idea here is that after the XNOR is performed, we next perform the full bit add. The sense amplifier can read the marker addresses that are stored in the buffer directly from the computational subarray. Hence why we can have all of this done as local memory operations. And it is using these marker table values in conjunction with the counts that are stored in the buffer as the operands for the full bit add, we can drastically increase our throughput. So here I mentioned the multiplexer, which is I'll highlight that specifically in the diagram I show next. But yeah, this translates to a carry ripple adder, which I'll show you right here. So the idea is we have this 128 columns, these 128 columns in the subarray, which can be sort of thought of in these 32-bit sequences that will end up being used for this add. So here you can see how this will how the process will kind of work. The second 128 rows contain all these marker table values. They will be accessed after the XNOR function by the multiplexer. So the multiplexer will choose one of these 4 32-bit sequences. And then there are going to be we have 32 12 transistor full bit adders that are all working sort of in conjunction with each other. Each one is assigned to one of these columns. The idea is that they're all operating simultaneously. So depending on the inputted nucleotide, the multiplexer will select one of these 32-bit sequences. All of the additions will run simultaneously, and then it'll move on to the next cycle. And it's because of this particular localized memory operation where we have these marker table addresses that correspond to the reference sequence from the Burroughs-Wheeler transform. These marker table values are stored in the same subarray that allows for us to drastically increase our throughput. So, right, so some details on how it was tested. So the circuit, it was all pretty much a simulation. They were both made in-house, so to speak. The circuitry was simulated by this kit inspector. It's a 45 nanometer technology process from one of the NCSU development kit libraries. The results of the circuitry simulation were fed into an architecture simulation that's based on NVMain. The idea behind this was that it could alter the configuration files based on this memory organization, so that they could kind of assess how this data partitioning was working, and it would give it would output the results. This was fed into a behavioral simulation that they made in MATLAB. This was how they were able to sort of gauge their energy and latency parameters. And these are the other primary accelerators that were compared. So ReCAM is kind of the only one that's like significantly different because it's implemented via dynamic programming. The rest are all similarly FM index acceleration solutions. Ambit I already touched on, DRISA, the one transistor, one capacitor model that is probably the most comparable in terms of performance to our architecture. Of course, we test on CPU and GPU, and this is implemented with SOAP 2 and SOAP 3. And of course, FPGA, which was discussed in the previous presentation too. So the reference genome that was used was HG19. This is just the reference with which we're aligning to, and all of the short reads were simulated by ART Simulator, and we had 10 million of them at 100 base pair read codes. Right, so this is what our results sort of look like. So our proposed, this is us here. We outperform in terms of giga operations per second, most notably with regard to the XNOR operation, because we can perform this in one cycle. There aren't any additional rows that must be copied. And sense amplification is shortened due to our the reliability of this 3-transistor XNOR gate. So yeah, we have, so in other words, the implementations on CPU and GPU were noticeably restricted. And it's due to this, von Neumann bottleneck. The processing memory solutions, which were those that were the FM index-based solutions, those perform significantly better. Among those, the best being our Aligner D algorithm. And this is how much it's outperforming them, just as a quick caption. Right, so the energy consumption is another important element. There's one of the most energy expensive operations is performing these additional row copies, especially when it comes to big data. Right, so because we only need to deal with one row at a time, because of our data partitioning, it is we consume significantly less energy relative to this sort of baseline, normalized to the baseline view with regard to the CPU. Right, so again, we're achieving a significantly higher throughput, and our power consumption is kind of in the same, kind of reflects the same sentiment. We're only hitting like 6.5 watts. It's getting it, but it's there's a lot more off-chip memory operations that are off-chip memory requests that are occurring in CPU, GPU, but the other processing and memory architectures are much closer to this. And then our throughput with respect to the power and power per unit area. Right, so again, we're significantly outperforming most of the other processing and memory architectures. Again, this is the only other architecture that is significantly or is notably closer. This is the only other model that was able to achieve an XNOR operation in 2 cycles. Excuse me. We achieve it in 1 cycle, they achieve it in 2 cycles, but 2 cycles is better than any of the other designs. So, right, and of course we're achieving much more area efficient design too, because we have a lot less transistors per column to perform our operations.
Strength of the paper removes row initialization overhead. Next, optimizes the algorithm, improves throughput and energy per operation compared to similar processing and memory architectures. Weaknesses mostly come from the fact that this has narrow functionality, so it focuses on XNOR and addition, and if we talk about broader logic, then it may not get similar gains. Then results are based on simulations, so they never fabricated an actual prototype, and performance benefits are demonstrated for FM index search and short read alignment, but also there is long read alignment. And for long read alignment, the applicability is not shown, and it is not supposed to do great for long read alignment. So the key takeaways: processing in memory is a promising direction to make read alignment much faster. 3-transistor XNOR can allow for very power efficient alignment. 12-transistor full adder significantly reduces energy consumption, and the updated algorithm takes advantage of all of that. Then for the future directions, there is this promising direction that is very similar to processing in memory, which is compute near memory. So we can place the data not in memory, but close to it. And also there is this pre-alignment filtering, which we've already seen. So this is an example of a paper that combines both approaches, pre-alignment filtering and the compute near memory thing, but also they do it for long reads, which is another direction, long read mapping, explore if that will, if mapping long reads will be more efficient. So there are these GPU accelerators for long reads, and also we can use this data compression, reference-based data compression. Instead of saving the entire sequence, just store the mismatching region, right? So there are papers on that. This is actually a preprint. It's very recent, 2025. Yep. So this is the end of our talk. So we can move to discussion now.
Well, first thing,
Yeah.
So trying to relax. That's equals also you see increments one base.
Can you please clarify what table you are referring to? Do you remember the slide number? No. The occurrence table, or we just have two tables, the occurrence table and the counts. Okay.
Additional one later.
Yeah. For the entire, for the Burr-Wheeler transform. For the sequence. Yeah. So the thing is, we showed you a very simple example where where the counts are basically the same as if we just listed them in alphabetical order. But if we take a real example when we have like multiple times every character is occurring, then the size of the table will enlarge. Right. We just take a bucket D, we just take a number D and just divide the table by D by simply taking every D, 1D, 2D, 3D. And the number can be whatever we want. If no questions, we can move to our questions.
Like the local area, how is the dollar sign? Calculated dollar and you're talking about that's wow.
I still don't understand the question.
saying how can you transfer?
Like
The
the
dollar
Burr,
sign goes first.
the dollar sign is something that we just put there to label the end of the string. Yeah, and then we just process, like we do the Burr-Wheeler transform as if it was the part of the string. Right.
It's the last. It's the last column of the Burroughs wheel.
Well, we can go to Burroughs-Wheeler algorithm again. Right. Okay, so we listed all suffixes, right? The first suffix starts at first position. I mean, index 0, then the next starts at index 1, right? Is that clear? So this way we build the suffix table. No, R is the read, and S is the sequence. So we are looking for this read and this. Yeah.
Then there are
We will have more, we will have a larger table. If the length of the sequence increases, we'll have more suffixes, we'll have more occurrences. Okay. Right. So this is what we got on the first step, right? Every suffix an index where it starts, right? So then we simply sort the suffixes in alphabetical order. So $sign goes first, and then the suffix that starts with A goes second, right? And then we just record the indices again. So this was 5, and 5 goes here. And then we record the first column for each suffix. This is the $sign. This starts with A. This starts with C. This starts with G. Is this step clear? And then we look at our first column that we got on the previous step. And we look at our sequence. What was the previous character before this $sign? The previous character was A. So we write this A in the last column. Yeah. This is the previous character for each of these characters in the first column. Right, correct. Right. 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 Yeah, great. Yeah.
I think it was. Okay, so we'll meet as we promised. I think someone forgot his laptop. Yeah. As we discussed, we will meet on Thursday at 5 o'clock for free talk. We'll try to take a group picture, so be ready. Right? Don't mess it. For those who will attend online, we'll try to have their picture here. Right? So yeah, thank you so much for staying until now. It's really a record. All right, we'll see you in a few days. Don't forget to send your vote as usual. This is true for everyone on Zoom and